Hello, everyone. Welcome back to another episode on Demystifying Medicine. Today, we'll be discussing live medicine. As the field of medicine grows larger and new antibiotics are being manufactured to combat ever-growing bacterial and fungal infections, many bacteria are beginning to evolve. They have developed mechanisms to resist commonly used antibiotics. This has become an urgent global public health threat, taking 1.27 million lives worldwide, with the highest count of 5 million deaths in 2019. Before we dive into the new discovery made, let's begin by defining what antibiotic resistance is. It is when germs, such as bacteria and fungi, develop the ability to survive against conventional antibiotics. Since they do not respond to these drugs and continue to multiply inside the body, it raises the problem in treating infections as they become resistant and the bacteria has become harder to kill. You must now be wondering, how is this even possible? Well, let's jump into the science behind this phenomenon. All organisms have cells, the basic unit of life. Held inside of these cells is our blueprint, or our DNA. Our DNA gives our body instructions on how to make us, us. DNA is replicated in order for cellular mechanisms to happen, such as creating more cells. DNA is composed of smaller units called nucleotides, and specific patterns of nucleotides code for specific proteins. So antibiotic resistance is caused when a mutation occurs where one of these nucleotides is incorrectly placed during the replication of bacterial DNA. Mutations lead to changes of protein structures, which in turn can have detrimental effects such as delayed development or impaired physiological structures with limited functions, but not all effects are negative. In this case, some mutations work in favor of the organisms, where a new pattern of bases can be translated into a new protein that can then provide the bacteria with a new defense mechanism against the drugs designed to kill them. Although this helps the bacteria to survive, this is not great news for humans suffering from infection. In fact, it can lead to serious consequences such as organ failure, prolonged care and recovery, increased rate of mortality, and worsening of disease caused by bacteria. In some cases, patients are left with no treatment options if the bacteria do not respond to any of them. This is particularly a problem when patients undergo surgery and doctors install medical implants inside their bodies. These implants are susceptible to causing infection due to resistant bacteria growing on them. However, a recent article uncovers a novel treatment option that is being developed to overcome antibiotic resistance. This is called live medicine. Researchers at the Center for Genomic Regulation and Pulmobiotics SL have modified and changed the structure and function of a common species of bacteria called Mycoplasma pneumoniae. Mycoplasma pneumoniae is primarily responsible for causing a chest cold, a type of atypical pneumonia, which is a milder version of actual pneumonia. The researchers removed the bacteria's ability to cause disease and reprogrammed it to attack other harmful bacteria on the surfaces of medical implants. This bacterium was chosen to be re-engineered due to its lack of cell wall, which is a rigid barrier that makes it hard for substances and proteins to move in and out of the cell. Since there is no cell wall in this bacterium, it will be easier for it to release therapeutic molecules from inside the cell. It works by attacking layers created by bacteria, known as a biofilm. Biofilm are colonies of bacteria that stick together on top of the surface of medical devices and create a barrier against common antibiotics. It also prevents the human immune system from destroying it. In an older study, the bacteria that the researchers tested their live medicine was against Staphylococcus aureus. This type of bacteria is the leading cause of skin and tissue infections. It is also one of the most common species that create a strong biofilm and does not respond to conventional antibiotics, making it a suitable test subject. For the purpose of this study and the Mycoplasma pneumoniae's ability to naturally adapt to the environment of the lungs, the live medicine was tested against Staphylococcus aureus biofilm growing around breathing tubes. Mycoplasma pneumoniae was reprogrammed to produce and efficiently release two different enzymes, which are proteins that dissolve the biofilm and then attack the cell walls of the bacteria, creating the biofilm. Due to its success, modified bacteria possess high potential as a treatment for other diseases. This is due to its ability to carry enzymes and therapeutic molecules to the region of disease of interest. They can offer continuous production of the therapeutic molecules inside the body and can be modified with different molecules that target other diseases. In a more recent study, researchers at the same institute used Mycoplasma pneumoniae against Pseudomonas aeruginosa. This type of bacteria is increasing the rate of hospital-acquired infections and has now become resistant to many types of antibiotics. It is also another bacteria that can create strong biofilms around breathing tubes. To test its efficacy, they administered its dose in mice with lung infections by using it in combination with low doses of antibiotics. The results yielded a significant reduction in the severity of infection in the mice lungs and a doubled survival rate. Not only did they test the modified bacteria against lung infections, but also tested its efficacy against biofilms from breathing tubes of patients in the ICU. Mycoplasma pneumoniae successfully penetrated the barrier of the biofilm and dissolved. Other treatment options include enzymes or antibodies, which are typically of high toxicity to normal human tissue and cells, but by localizing enzymatic activity of the medication to only the area that is infected by the bacteria, it is possible to treat the infection without harming the surrounding tissue. As previously stated, we can see that live medicine holds strong potential, and it may just be the medical innovation that the world desperately needs as antibiotic resistance is on the rise.
Created using Powtoon.